Hi class, this is your instructor, Skylar Huff. We're now in Chapter 10, How Cells Divide. All species of organisms, bacteria, alligators, the weeds in a lawn, which I say weeds are great in a lawn. In fact, I may need to come up with a better name for those other than weeds. Just call them native plants, if there are native plants. Otherwise, they could be non-native plants or invasive plants. But they all grow and reproduce. From single-celled organisms to complex multicellular organisms like you yourself and even me, all species produce offspring like themselves and pass on hereditary information that makes them what they are. In this chapter, we will examine how cells like the white blood cell shown in your text and now shown to you here divide and reproduce. Cell division is necessary for the growth of organisms, for wound healing, and to replace cells that are lost regularly, such as those in your skin and in the lining of your gut, not to mention lining of your mouth. The mechanism of cell reproduction and its biological consequences have changed significantly during evolution of life on Earth. The process is complex in eukaryotes, involving both the replication of chromosomes and their separation into daughter cells. Much of what we're learning about the causes of cancer relates to how cells control this process, and in particular, their tendency to divide, a mechanism that in broad outline remains the same in all eukaryotes. In this chapter, we'll go through these 7 sections outlined here. So let's go ahead and start. Bacteria divide as a way of reproducing themselves. Although bacteria exchange DNA, they do not have a sexual cycle like eukaryotes. Thus, all growth in bacterial population is due to division to produce new cells. The reproduction of bacteria is clonal. That is, each cell produced by cell division is an identical copy of the original cell. They're clones. Cell division in both bacteria and eukaryotes produces 2 new cells with the same genetic information as the original. Despite the differences in these cell types, the essentials of the process are the same: duplication and segregation of genetic information into daughter cells, and the division of cellular contents. We begin by looking at the simpler of 2 processes, binary fission, which occurs in bacteria. Most bacteria have a genome made up of a single circular DNA molecule. In spite of this apparent simplicity, the DNA molecule of the bacterium Escherichia coli, known as E. coli, is actually on the order of 500 times longer than the cell itself. Thus, this, in quotations, simple structure is actually packaged into very tightly to fit into the cell. And you're kind of seeing that picture of binary fission now. Although not found in a nucleus, the DNA is located in a region called the nucleoid that is distinct from the cytoplasm around it. As you can see maybe here at the top, the nucleoid region, no nucleus. The compaction and organization of the nucleoid involves a class of proteins called structural maintenance of chromosome, or SMC proteins. These are ancient proteins that have diversified over evolutionary time to fulfill a variety of roles related to DNA organization in different lineages. In eukaryotes, the cohesin and condensin proteins discussed throughout this chapter are SMC proteins. During binary fission, the chromosome is replicated and the 2 products are partitioned to each end of the prior cell prior to the actual division of the cell. One key feature of bacterial cell division is that replication and partitioning of the chromosome occur as a concerted process. In contrast, DNA replication in eukaryotes, or as in eukaryotic cells, occurs early in division, and the chromosome separation occurs much later. Binary fission begins with replication of the bacterial DNA at a specific site, the origin. So this will be introduced to you all in Chapter 14, and proceeds in both directions around the circular DNA to a specific site of termination. So if you look at this figure called Figure 10.1, you shall see in green the origin, and then kind of there in red where things shall end, the termination. The cell grows by elongation, and division occurs roughly at mid-cell. The cell grows by elongation, and division occurs at roughly mid-cell. So for many years, it was thought that newly replicated E. coli DNA molecules were passively segregated by attachment to and growth of the membrane as the cell elongated. Experiments that followed the movement of the origin of replication show that it is at this mid-cell prior to replication. Then the newly replicated origins move toward opposite ends of the cell, and then this movement is faster than the rate of elongation, showing that this growth alone is not enough. The origins appear to be captured at the 1/4 and 3/4 positions relative to the length of the cell, which will be mid-cell of the resulting daughter cells. Although the actual mechanism of chromosome segregation is unclear, the order of events is not: during replication, the first origin, then the rest of the newly replicated chromosomes are moved to opposite ends of the cell as two new nucleoids are assembled. The final event of replication is decatenation or untangling of the final replication products. After replication and segregation, the mid-cell region is cleared of daughter nucleoids and the division occurs. The force behind the chromosome segregation has been attributed to DNA replication itself, transcription, and the polymerization of actin-like molecules. At this point, no single model appears to explain the process, and it may involve more than one. The cell's other components are partitioned by the growth of the new membrane and the production of the septum, as you are now seeing in front of you, here right above number 4. And this process, termed septation, usually occurs at the midpoint of the cell. It begins with the formation of a ring composed of many copies of the protein FTSZ, and then next, the accumulation of a number of proteins occurs, including ones embedded in the cell membrane. The structure contracts inward radially until the cell pinches off into two new cells. The mid-cell location of the FTSZ ring is caused by factors at the poles that inhibits the FTZ ring formation. The FTZS protein is found in most prokaryotes, including archaea. It can form filaments and rings, and three-dimensional crystals show a high degree of similarity to the eukaryotic tubulin. However, its role in bacterial division is quite different from the role of tubulin in mitosis and eukaryotes. The evolution of eukaryotic cells included much more complex genomes composed of multiple linear chromosomes housed in a membrane-bound nucleus. These complex genomes may be possible due to the evolution of mechanisms that delay chromosome separation after replication. Although it is unclear how this ability to keep chromosomes together evolved, it does seem more closely related to binary fission than we once thought. So there you have it. Chromosomes were first observed by the German embryologist Walter Flemming, and this was in 1879 while he was examining the rapidly dividing cells of salamander larvae. Using what would now be a rather primitive light microscope, Flemming saw minute threads within their nuclei that appeared to be dividing lengthwise. Flemming called their division mitosis, based on the Greek word mitos, meaning thread. Since their initial discovery, chromosomes have been found in the cells of all eukaryotes, although their numbers vary enormously between different species. Organisms have been found with only a single pair of chromosomes, and some ferns with more than 500 pairs. You can see table 10.1 to show you groups of organisms with their total number of chromosomes. Most eukaryotes have between 10 and 50 chromosomes in their body cells. The defining number of different chromosomes in a species when defining the number of different chromosomes in a species, geneticists count the haploid number of chromosomes. This refers to one complete set of chromosomes necessary to define an organism. For humans and many other species, the total number of chromosomes in a cell is called the diploid number, which is twice the haploid number. For humans, the haploid number is 23, and the diploid number is 46. Diploid chromosomes represent the equal genetic contribution that each parent makes to offspring. We refer to the maternal and paternal chromosomes as being homologous or homologous chromosomes, and each one of the pair is termed a homologue. So each parent provides a homologue to make up the pair known as homologous chromosomes. Chromosomes vary in size, staining properties, the location of a visible constricted region called the centromere, and the relative length of the two arms on either side of the centromere. And to kind of help show this, I'm going to go back to figure 10.4, and there you have what I'm referring to now. The particular array of chromosomes an individual organism possesses is called a karyotype, and it is now shown here in figure 10.5. So figure 10.5 shows a normal human karyotype. And there you have that 23rd pair of chromosomes, which is what determines sex. So in this case, this would be from a male. It is amazing that after more than 100 years and a variety of increasing sophisticated methods of analysis, we still do not know the precise structure of chromosomes. The details of how this structure changes over the course of the cell cycle is not known either. What has become clear is that the nucleus is much more organized than originally conceived. Chromosomes are composed of chromatin, a complex of DNA and protein, usually about 40% DNA and 60% protein. A significant amount of RNA is also associated with chromosomes because chromosomes are the sites of RNA synthesis. Each chromosome contains a single uninterrupted DNA molecule. A typical human chromosome contains about 140 million nucleotides in its DNA. If you were to stretch the DNA strand in a human chromosome, the average length is about 4.3 centimeters, which is roughly 1.7 inches. Well, exactly 1.7 inches, excuse me. Fitting this length of DNA into a cell nucleus is like cramming a string the length of a football field into a baseball, and that's only one chromosome. In a cell, however, the DNA is compacted, allowing it to fit into a much smaller space, and the level of compaction changes with the cell cycle as needed to move chromosomes about the cell. If we gently disrupt a eukaryotic nucleus and examine the DNA with an electron microscope, we find that it resembles a string of beads. Let's find some beads. Here are the beads. The beads are a structure we call the nucleosome. And the nucleosome, which is composed of DNA and histone proteins. So histones have a positive charge because of an abundance of the basic amino acids arginine and lysine. Thus, they are strongly attracted to negatively charged DNA. Histone proteins are some of the most highly evolutionarily conserved proteins known, indicating both their age and their importance. Nucleosomes contain four primary histones, H2A, H2B, H3, and H4. These associate together to form a large complex containing two of each core histone to form an octamer, which acts as a spool that DNA is wound around. Each histone octamer has 147 base pairs of DNA wrapped in 1.7 left-hand turns to form the nucleosome core particle, which has a diameter of about 10 nanometers. So each of these is separated by a variable length of DNA of 20 to 80 base pairs called linker DNA. thus the beads on a string appearance. And you can see that in figure 10.6, which I showed moments ago. An additional histone, H1, can interact with the linker DNA, and the core particle histones also have protruding tails that are subject to post-translational modifications, which facilitate interactions between nucleosomes. If isolated DNA wrapped in nucleosomes is incubated under low salt conditions with histone H1, it will form an even more compact structure with a diameter of 30 nanometers. This so-called 30 nanometer fiber is formed by either coiling into a spiral called a solenoid or forming a zigzag structure with the same diameter. The 30 nanometer fiber was long considered the usual state of chromatin in non-dividing cells, but there is little actual evidence that this fiber exists in cells. So starting at the top left and go clockwise. The top row shows the organization of chromatin in interphase in the nucleus. The individual chromosomes tend to be organized into territories that exclude other chromosomes. Within a chromosome, chromatin is organized into compartments that consist of either active or inactive regions. These in turn are formed by looped structures called TADs. The base of a loop is anchored by cohesin and CTCF. The DNA in TADs is organized into nucleosomes, and this, which consists of DNA wrapped around a core particle of 8 histone proteins. So let's speak to those TADs. New imaging techniques combined with high throughput methods to assess interactions between genomic regions have revealed the organization of chromosomes in the eukaryotic nucleus. Chromatin is organized into territories which contain individual chromosomes, and these are further organized into compartments within each chromosome. So at the highest level of resolution, genomic regions interact to form topologically associated domains. The TADs I spoke to earlier, they are referred to as topologically associated domains. These topologically associated domains involve loops of DNA in nucleosomes that are anchored at their base by 2 proteins, a DNA binding protein called CTCF and the cohesin complex of proteins. The loops may be involved in the interactions between nucleosomes, and this organization may also affect the control of gene expression by bringing relatively distant regions close together and keeping other regions compacted. Geneticists have recognized for years, though, that domains of chromatin called heterochromatin were not active, and other domains of chromatin called euchromatin are active. These names originally were based on the staining patterns seen in the light microscope, but it now appears that this may extend to the kinds of organization found in the compartments of topologically associated domains, with some being more condensed and inactive, and others more open and active. So there you have it. The state of chromosome condensation changes through the cell cycle. Between divisions, chromatin is relatively diffuse, then increasingly condensed to allow the separation of chromosomes during cell division, then condensed again after division. The mental image most of us have of a chromosome, if we have one at all, is of the highly condensed X shape metaphase chromosome. This structure has been shown to be formed by organizing DNA on a protein scaffold. The scaffold was first visualized by removing histones from isolated metaphase chromosomes, which caused the DNA to form a massive tangle of spaghetti-like fibers of DNA surrounding an X-shaped scaffold of proteins. For many years, the domain model of successive coiling to form the 30 nanometer fiber, which was further condensed by radial looping from this scaffold of proteins. While models of this type have not been completely rejected, the only part with strong experimental support is the existence of protein scaffold to organize chromatin. The analysis of genomic interactions reveals some interesting patterns over the course of the cell cycle. Topologically associated domains seen in the interphase cells disappear during prophase, and more regularly interactions occur along a chromosome. So this coincides with the replacement cohesin by a protein of condensin proteins. Condensin and cohesin are both structural maintenance chromosome proteins that interact with DNA and appear to be able to induce loops in DNA. This does not appear to involve the 30 nanometer fiber, with DNA appearing to be more fluid, with nucleosome interacting to form dense networks. One new model envisions condensin proteins organized along the scaffold using energy from ATP hydrolysis to form regular loops of DNA in nucleosomes. This is then twisted over the course of the cell cycle to further compact loops, with the number of loops also increasing with progress through prophase. In this model, DNA is wrapped into nucleosomes, and then interactions of linker histones and nucleosomes themselves can change the density of liquid DNA. While this might sound bizarre, it fits much of the new DNA on the chromosome conformation. When DNA is replicated, the single molecule in each chromosome becomes two identical DNA molecules held together at their centromere by cohesins. As each chromosome becomes more condensed, it becomes visible as the X-shaped structure in a karyotype. At this point, we still call this one chromosome because it is composed of two sister chromatids. So upon replication, though you have each chromosome that has become more condensed and visible as an X-shaped structure, we still call it a chromosome. And in prior times, I've referred to it as being a duplicated chromosome, but it's still a chromosome with two sister chromatids. You can see that here in figure 10.7. So we have homologous chromosomes there on the left, and then you have, again, homologous chromosomes there on the right, and that is upon replication. The fact that the products of replication are held together is critical to the division process. At the end of the division process, each cell must have a complete set of chromosomes. If we were designing a system, we might use some kind of label to identify each chromosome, much like labels most of us use when we duplicate files on a computer. Instead of labeling chromosomes, the cell glues the replication products together at the centromere. The process of mitosis then separates all of these copies at the same time, ensuring that each daughter cell gets one copy of each chromosome. Compared with prokaryotes, the increased size and more complex organization of eukaryotic genomes requires radical changes in the partitioning of replicated genomes into daughter cells. The cell cycle requires the duplication of the genome, its accurate segregation, and the division of cellular contents. The cell cycle is divided into phases based on the key events of genome duplication and segregation. The cell cycle is usually diagrammed as in figure 10.8. Figure 10.8. We have G1 or gap 1 or gap phase 1, shown there in yellow. We have S for synthesis, shown there in red. We have G2 or gap 2 or gap phase 2, shown in green. We then, and these make up what is known as interphase. Let me say that. These G1, S, and G2 are what make up interphase. Then there is what is called M phase or mitosis, including, I'll just bluntly say prophase, metaphase, anaphase, and telophase or telophase, which thereafter is cytokinesis, which I might sometimes link with telophase. And this is the cell cycle. G1 is the primary growth phase of the cell cycle. The term gap refers to its filling the gap between cytokinesis and DNA synthesis. For most cells, this is the longest phase. S. This is the phase in which the cell synthesizes a replica of the genome, so replication occurs. G2 is the second growth phase and preparation for separation of newly replicated genome. This phase fills the gap between DNA synthesis and the beginning of mitosis. During this phase, microtubules begin to reorganize to form a spindle. These make up what is known as interphase, the portion of the cell cycle between cell divisions. Mitosis is the phase of the cell cycle in which the spindle apparatus assembles, binds the chromosomes, and moves the sister chromatids apart. Mitosis is the essential step in the separation of two daughter genomes. It is traditionally subdivided into 5 stages. I might divide it into 4. Prophase, prometaphase, metaphase, anaphase, and telophase, and then cytokinesis. This is the phase of the cell cycle when the cytoplasm divides, creating 2 daughter cells. In animal cells, the microtubule spindle helps position the contracting ring of actin that constricts like drawing a string to pinch the cell into 2. In cells with a cell wall, such as plant cells, a cell plate forms between the dividing cells. Mitosis and cytokinesis together are usually referred to collectively as M phase, as I mentioned earlier, to distinguish the dividing phase from interphase. And I will likely refer to it as such as being interphase and M phase or mitosis, which includes cytokinesis. The time it takes to complete the cell cycle varies greatly. Cells in animal embryos can complete their cell cycle in under 20 minutes. The shortest known animal nuclear division cycle occurs in the fruit fly in roughly 8 minutes. These cells simply divide their nuclei as quickly as they can replicate their DNA without cell growth. Half of the cell cycle is taken up by S and then half by M, and essentially none by G1 or G2. Because mature cells require time to grow, most of their cycles are much longer than those of embryonic tissue. Typically, a dividing mammalian cell completes the cell cycle in about 24 hours. But some cells, such as certain cells in the human liver, have cell cycles lasting more than a year. During the cell cycle, growth occurs throughout G1 and G2 phases, as well as the S phase. The M phase takes only about an hour, a small fraction of the entire cell cycle. So most of the variation in the cell cycle between organisms or cell types occurs in G1 phase. Cells often pause in G1 before DNA replication and enter a resting state called G0 phase. Cells may remain in this phase for days to years before resuming cell division. At any given time, most cells in an animal's body are in G0 phase. Some, such as muscle and nerve cells, remain there permanently. Others, such as liver cells, can resume G1 phase in response to factors released during injury. The cell grows throughout interphase. The G1 and G2 segments of interphase are periods of active growth during which proteins are synthesized and cell organelles are produced. The cell's DNA is replicated during S phase of the cell cycle. The events that occur during interphase are important for the successful completion of mitosis. During G1, cells undergo the major portion of their growth. During S phase, each chromosome replicates to produce 2 sister chromatids, which remain attached to each other at the centromere. In G2 phase, the chromosomes begin to condense. After S phase, the sister chromatids appear to share a common centromere, but at the molecular level, the DNA of the centromere has actually already been replicated, so there are 2 complete DNA molecules. These 2 chromatids are held together by cohesin proteins at the centromere, and each chromatid has its own set of kinetochore proteins. In multicellular animals during G2, most of the cohesins along the arms that held sister chromatids together after replication are replaced by condensins as chromosomes begin to condense. So this is showing you figure 10.9, where you see that each chromatid has its own set of kinetochore proteins, shown there in the center, near the centromere, opposite sides. The action of condensin is required for later stages of chromosome condensation. This leaves chromosomes still attached tightly at the centromere, but loosely attached elsewhere. So the proteins are here found at the centromere. So what you're seeing in this is a human mitotic chromosome that has been stained for condensin, which is in blue, cohesin in green, and the centromere-specific histone CENPA in red. The cohesin proteins hold the centromeres of sister chromatids together. Another significant alteration to the cellular architecture takes place during Ch10 phase. The usual organization of the cytoskeleton microtubules is disassembled and begins to be rearranged to assemble the machinery needed to move chromosomes to opposite poles of the cell. In animal cells, a pair of microtubules organizing centers called centrioles replicate, producing one for each pole. All eukaryotic cells undertake an extensive synthesis of tubulin, the protein that forms microtubules that will be used to construct the mitotic spindle. The process of mitosis is one of the most dramatic and beautiful biological processes that we can easily observe. In our attempts to understand this process, we have divided into discrete phases, but it should be remembered that this is a dynamic, continuous process, not a set of discrete steps. This process is shown both schematically and in micrographs in figure 10.11. You're seeing that same figure before yourself now. This is interphase Ch10. When the chromosome condensation initiated in Ch10 reaches the point at which individual condensed chromosomes first become visible with the light microscope, the first stage of mitosis, prophase, has begun. The condensation process continues throughout the prophase. Consequently, chromosomes start prophase as minute threads appear quite bulky before its conclusion. The spindle apparatus will later separate the sister chromatids forms during prophase. The spindle apparatus that will later separate the sister chromatids forms during prophase. The spindle replaces the normal microtubule structure disassembled in the Ch10 phase. In animal cells, the centriole pair formed during Ch10 phase begin to move apart early in prophase, forming between them an axis of microtubules that make up the spindle. This consists of polar microtubules which emanate from each pole and overlap at the mid-cell, and kinetochore microtubules which attach to each chromosome at its kinetochore. By the time the centrioles reach the opposite poles of the cell, they have established a bridge of microtubules. In plants, a similar bridge of microtubular fibers form between opposite poles, although centrioles are absent in plant cells. In animal cell mitosis, the centrioles extend in a radial array of microtubules toward the nearby plasma membrane when they reach the poles of the cell. This arrangement of microtubules is called an aster. Although aster's function is not fully understood, it probably braces the centrioles against the membrane and stiffens the point of microtubule attachment during the retraction of the spindle. Plant cells, which have rigid cell walls, do not form asters. During formation of the spindle apparatus, the nuclear envelope breaks down and the endoplasmic reticulum reabsorbs its components. At this point, the microtubule spindle fibers extend completely across the cell, from one pole to the other. Their orientation determines the plane in which the cell will subsequently divide through the center of the right angles to the spindle apparatus. This is prophase. The transition from prophase to prometaphase occurs following the disassembly of the nuclear envelope. During prometaphase, the condensed chromosomes become attached to the spindle by their kinetochores. Each chromosome possesses two kinetochores, one attached to the centromere region of each sister chromatids. Prometaphase. As prometaphase continues, a second group of microtubules grows from the poles of the cell toward the centromeres. These microtubules are captured by kinetochores on each pair of the sister chromatids. This results in kinetochores of each sister chromatid being connected to opposite poles of the spindle. This bipolar attachment is critical to the process of mitosis. Any mistakes in microtubule positioning can be disastrous. For example, the attachment of kinetochores on both sister chromatids to the same pole leads to failure of sister chromatid separation. They'll be pulled to the same pole, ending up in the same daughter cell, with the other daughter cell missing that chromosome. So each chromosome is attached to the spindle by microtubules running from opposite poles to the kinetochores by sister chromatids. The chromosomes are being pulled simultaneously toward each pole, leading to a jerky motion that eventually pulls all of the chromosomes to the equator of the cell. At this point, the chromosomes are arranged at the equator with sister chromatids under tension and oriented toward the poles by their kinetochore microtubules. The force that moves the chromosomes has been of great interest since the process of mitosis was first observed. Two basic mechanisms have been proposed to explain this. One is assembly and disassembly of microtubules provides the force to move chromosomes. And two, motor proteins located at the kinetochore and the poles of the cell pull on the microtubules to provide force. Data have been obtained to support both mechanisms. In support of the microtubule shortening proposal, isolated chromosomes have been pulled by microtubule disassembly. The spindle is a very dynamic structure with microtubules being added to the kinetochore and shortened at the poles, even during metaphase. In support of the motor protein proposal, multiple motor proteins have been identified as kinetochore proteins, and inhibition of the motor protein denine slows chromosome separation at anaphase. Like many phenomena we analyze in living systems, the answer is not a simple either-or choice. Both mechanisms probably at work. The alignment of the chromosomes in the center of the cell signals the third stage of mitosis, metaphase. When viewed with a light microscope, the chromosomes appear to array themselves in a circle at the inner circumference of the cell, just as the equator girdles the earth. Okay. And you're seeing that right now in figure 10:12 here, and even with figure 10:11 there. An imaginary plane perpendicular to the axis of the spindle that passes through the circle is called the metaphase plate. The metaphase plate is not an actual structure, but rather an indication of the future axis of cell division. Positioned by the microtubules attached to the kinetochores of the centromeres, all the chromosomes line up at the metaphase plate. At this point, their centromeres are neatly arrayed in a circle, equidistant from the two poles of the cell, with microtubules extending back toward opposite poles of the cell. The cell is prepared to properly separate sister chromatids such that each daughter cell will receive a complete set of chromosomes. Thus, metaphase is really a transitional phase in which all the preparations are checked before the action continues. Of all the stages of mitosis shown in Figure 10.11, anaphase is the shortest and the most amazing to watch. It begins when the proteins holding sister chromatids held together at the centromere are removed. Up to this point in mitosis, sister chromatids have been held together by cohesin proteins concentrated at the centromere. The key event in anaphase, then, is the simultaneous removal of these proteins from all of the chromosomes. The control and details of this process are going to be discussed a bit later. Free from each other, the chromosomes, no longer sister chromatids, are pulled rapidly toward the poles to which their kinetochores are attached. In this process, two forms of movement take place simultaneously, each driven by microtubules. These movements are often called anaphase A and anaphase B to distinguish them. During anaphase A, the kinetochores are pulled toward the poles, as microtubules that connect them to the poles shorten. This shortening process is not a contraction. The microtubules do not get any thicker. Instead, the tubulin subunits are removed from the kinetochore ends of the microtubules. As more subunits are removed, the chromatid-bearing microtubules are progressively disassembled, and the chromatids are pulled ever closer to the poles of the cell. Second, during anaphase B, the poles move apart as microtubular spindle fibers physically anchored to opposite poles slide past each other away from the center of the cell. And this is shown to you now in Figure 10.13. Because another group of microtubules attaches the chromosomes to the poles, the chromosomes move apart too. If a flexible membrane surrounds a cell, it becomes visibly elongated. When the chromosomes separate in anaphase, the accurate partitioning of the replicated genome, the essential element of mitosis, is complete. In telophase, the spindle apparatus disassembles as the microtubules are broken down into tubulin monomers that can be used to construct the cytoskeletons of the daughter cells. A nuclear envelope forms around each set of sister chromatids, which can now be called chromosomes because they are no longer attached at the centromere. The chromosomes soon begin to uncoil into the more extended form that permits gene expression. One of the early group of genes expressed after mitosis is comprises the rRNA genes. So again, one of the early group of genes expressed after mitosis is complete, comprises the rRNA genes, resulting in the reappearance of the nucleolus. Telophase can be viewed as a reversal of the process of prophase, bringing the cell back to the state of interphase. Mitosis is complete at the end of telophase. The eukaryotic cell has partitioned its replicated genome into two new nuclei, positioned at opposite ends of the cell. Other cytoplasmic organelles, including the mitochondria and chloroplasts, if present, were reassorted to areas that will separate and become the daughter cells. Cell division is still not complete at the end of mitosis. However, because the division of the cell body properly has not begun, the phase of the cell cycle when the cell actually divides is called cytokinesis. It generally involves the cleavage of the cell into roughly equal halves. In animal cells and cells of other eukaryotes that lack cell walls, cytokinesis is achieved by means of constricting a belt of actin filaments. As these filaments slide past one another, the diameter of the belt decreases, pinching the cell, creating a cleavage furrow around the cell's circumference. As constriction proceeds, the furrow deepens until it eventually slices all the way into the center of the cell. At this point, the cell is divided into two. So in Figure 10.14a, the cleavage furrow around a dividing frog egg, and in Figure 10.14b, the completion of cytokinesis in an animal cell. The two daughter cells are still joined by a thin band of cytoplasm, largely by microtubules. Plant cell walls are too far rigid to be squeezed into by actin filaments. Instead, these cells assemble membrane components in their interior at right angles to the spindle apparatus. This expanding membrane partition, called the acell plate, continues to grow outward until it reaches the interior surface of the plasma membrane and fuses with it, effectively dividing the cell into two. So this now shows that in figure 10.15. Cellulose is then laid down on the new membranes, creating two new cell walls. The space between the daughter cells becomes impregnated with pectins and is called a middle lamella. In most fungi and some groups of protists, the nuclear membrane does not dissolve, and as a result, all the events of mitosis occur entirely within the nucleus. Only after mitosis is complete in these organisms does the nucleus divide into two daughter nuclei. Then during cytokinesis, one nucleus goes into each daughter cell. This separate nuclear division phase of the cell cycle does not occur in plants, animals, or most protists. After cytokinesis in any eukaryotic cell, the two daughter cells contain all the components of a complete cell, whereas mitosis ensures that both daughter cells contain a full complement of chromosomes. No similar mechanism ensures that organelles such as the mitochondria and chloroplasts are distributed equally between the daughter cells. But as long as at least one of each organelle is present in each cell, the organelles can replicate to reach the number appropriate for that cell. Controlling the process of cell division is critical. In multicellular organisms like humans, loss of this control leads to the disease cancer. Biologists have approached this problem from a variety of disciplines and perspectives. This has uncovered factors that drive the cell cycle forward and other factors that tend to rein in the process of division. There are two irreversible steps in the cell cycle: DNA replication and the separation of the sister chromatids. These 2 points are tightly controlled as other specific points we call checkpoints. At these checkpoints, the process can be assessed for accuracy and halted if there are errors. This leads to very high fidelity for the overall process. This also allows the cell cycle to respond to both internal and external signals, which are integrated at major checkpoints. The history of investigation into the control of the cell cycle is instructive in two ways. It first allows us to place modern observations into context, and secondly, we can see how biologists using very different approaches end up at the same place. A brief history follows, with 3 observations, then shows how they can be integrated into a single mechanism. Research on the activation of frog oocytes led to the discovery of a substance that was first called maturation promoting factor, or MPF. Frog oocytes, which go on to become egg cells, become arrested near the end of their development at G2 before meiosis I, which is the division leading to the production of gametes. That's our next chapter. They remain in this arrested state and await hormonal signaling to complete this division process. Cytoplasm taken from a variety of actively dividing cells could prematurely induce cell division when injected into oocytes, which you all can see in neither of those figures. I thought the figure was there, excuse me. These experiments indicated that the presence of a positive regulator of the cell cycle progression in the cytoplasm of dividing cells, MPF. These experiments also fit well in the cell cycle fusion experiments done with mitotic interphase cells that also indicated that cytoplasmic positive regulator could induce mitosis. Further studies highlighted 2 key aspects of maturation promoting factor. First, MPF activity was varied during the cell cycle, low in early G2, rising throughout the phase, then peaking in mitosis. Second, enzymatic activity of MPF involved the phosphorylation of proteins. This second point is not surprising given the importance of phosphorylation as a reversible switch on the activity of proteins. The first observation indicated that MPF itself was not always active, but rather being regulated with the cell cycle. And the second showed the possible enzymatic activity of MPF. Other researchers examined proteins produced during the cell cycle division in sea urchin embryos. They identified proteins that were produced in synchrony with the cell cycle named cyclins. These observations were extended in another marine invertebrate, the surf clam. Two forms of cyclin were found that cycled at slightly different times, reaching peak at G1S and at G2M boundaries. Despite much effort, no identified enzymatic activity was associated with these proteins. Their hallmark was the timing of their production and not any intrinsic activity. Geneticists using two different yeasts, budding yeast and fission yeast, as model systems set out to determine the genes necessary for control of the cell cycle. By isolating the mutants that were halted during division, they identified genes that were necessary for the cell cycle progression. These studies indicated that in yeast there were two critical control points: the commitment to DNA synthesis, called START, as it meant to committing to divide, and the commitment to mitosis. One particular gene, named CDC2, from fission yeast, was shown to be critical for passing both of these boundaries. All of these findings came together in an elegant fashion with following observations. First, a protein encoded by the CDC2 gene was shown to be a protein kinase. Second, the purification and identification of MPF showed that it was composed of both a cyclin component and a kinase component. Lastly, the kinase itself was the CDC2 protein. The CDC2 protein first identified cyclin-dependent kinase, or CDK, that is, a protein kinase enzyme that is only active when complexed with cyclin. This finding led to the renaming of MPF as mitosis promoting factor, as its role was clearly more general than simply promoting the maturation of frog oocytes. These CDK enzymes are the key positive drivers of the cell division cycle. They are often called the engine that drives cell division. The control of the cell cycle in higher eukaryotes is much more complex than the simple division of yeast, but the yeast model remains a useful framework for understanding more complex regulation. The discovery of CDKs and their role in the cell cycle is an excellent example of the progressive nature of science. Although we divide the cell cycle into two phases and further subdivide mitosis, cells actively control only 3 points in the cell cycle we call checkpoints: G1S, G2M, and late metaphase, the spindle checkpoint. These checkpoints allow the cell cycle to be delayed or halted when necessary. The cell uses these checkpoints to both assess its internal state and integrate external signals. Passage through these checkpoints is controlled by the CDK enzymes just described. The G1S checkpoint is the primary checkpoint at which the cell decides whether or not to divide. This checkpoint is therefore the primary checkpoint at which external signals can influence the events of the cycle. It is the phase during which growth factors affect the cell cycle and also the phase that links cell division to cell growth and nutrition. G1S, shown there in yellow. The G2M checkpoint has received a large amount of attention because of its complexity, and it's important as the stimulus for the events of mitosis. Historically, CDKs active at this checkpoint were first identified as maturation promoting factors, a term that has now evolved into M phase promoting factor, still MPF. Passage through this checkpoint represents the commitment to mitosis. This checkpoint assesses the success of DNA replication and can stall the cell cycle if DNA has not been accurately replicated. DNA damaging agents result in arrest at this checkpoint as well as at the G1S checkpoint. This is G2M shown in green. The spindle checkpoint ensures that all of the chromosomes are attached to the spindle in preparation for anaphase. The second irreversible step in the cell cycle is the separation of the chromosomes during anaphase, and therefore it is critical that they are properly arrayed at the metaphase plate. Spindle checkpoint here shown in purple. The primary molecular mechanism of the cell cycle control is phosphorylation, which you may recall is the addition of a phosphate group to the amino acids serine, thymine, tyrosine proteins. These enzymes accomplish this phosphorylation are the CDKs. So those are the enzymes that accomplish the phosphorylation, the cyclin-dependent kinases. The first important cell cycle kinase was identified in fission yeast named CDC2, now also called CDK1. In yeast, CDK can partner with different cyclins at different points in the cell cycle. Even in the simplified cell cycle of yeast, we are left with the important question of what controls the activity of the CDKs during the cell cycle. As the G1S checkpoint is approached, triggering the signal in yeast appears to be the accumulation of G1 cyclins. These form a complex of CDC2 to create the active G1S CDK, which phosphorylates a number of targets that bring about the increased enzyme activity for DNA replication. There you have it. MPF and its role in the G2M checkpoint has been extensively analyzed in a number of different experimental systems. The control of MPF is sensitive to agents that disrupt or delay replication and to agents that damage DNA. It was once thought that MPF was controlled solely by the level of M-phase specific cyclins, but it has now become clear that this is not the case. Although M-phase cyclin is necessary for MPF function, activity is controlled by an inhibitory phosphorylation of kinase component CDC2. The critical sign in the process is the removal of the inhibitory phosphates by a protein, phosphatase, and this action forms the molecular switch based on positive feedback because the MPF further activates its own activating phosphatase. The checkpoint assesses the balance between kinase that adds inhibitory phosphates with phosphatase that removes them. Damaged DNA acts through a complex pathway that includes damage sensing and a response to tip the balance toward inhibitory phosphorylation of MPF. Anaphase promoting complex, APC or APCC. The presence of all chromosomes at the metaphase plate and the tension of the microtubules between opposite poles are both important. The signal is transmitted through anaphase promoting complex, which is also called cyclosome. So the function of APCC is to trigger anaphase itself. The APC does not directly act on cohesin, but rather acts by marking a protein called securin for destruction. The securin protein acts as an inhibitor of another protease called separase that is specific for one component of the cohesin complex. Once inhibition is lifted, separase destroys cohesin. The APC has two main roles in mitosis. It activates the protease that removes cohesins holding the sister chromatids together, and it is necessary for the destruction of the mitotic cyclins to drive the cell out of mitosis. The APCC complex marks proteins for destruction by the proteasome, the organelle responsible for controlled degradation of proteins. The signal to degrade protein is the addition of a molecule called ubiquitin, and APCC acts as a ubiquitin ligase. So as we learn more about APCC and its functions, it is clear that the control of its activity is a key regulator of the cell cycle. So now that we've gone through those, let's now get to genetics and cancer. The unrestrained, uncontrolled growth of cells in animals leads to disease called cancer. Cancer is, in essence, a growth disorder of cells. It starts when an apparently normal cell begins to grow in an uncontrolled way. The result is a cluster of cells called a tumor that constantly expands in size. The clustered pink lung cells that are showed in figure 10.22 and not included in your text shows it, but they have begun to form a tumor. Malignant tumors are invasive. Their cells can spread from one original tumor to other areas of the body in a process called metastasis. Or a form of metastatic cancer. Anything that damages DNA, such as environmental factors like the chemicals in cigarette smoke, and I'll say vaping, or UV radiation and sunlight, can lead to mutations. Random mutations also arise with each round of DNA replication, many of which I would think are de novo and have no effect. If a gene is mutated that encodes for one of the proteins involved in controlling cell division, this can lead to uncontrolled growth. The search for cancer-causing genes produced two distinct classes of genes, oncogenes and tumor-suppressing genes. Oncogenes were discovered in viruses that cause cancer. The first being the SRC gene, which is carried by the Rous sarcoma virus. When oncogenes are introduced to cells, they cause malignant transformation. The first tumor-suppressing gene was discovered as the cause of the familiar cancer retinoblastoma. We will consider the similarities and differences between these two types of cancer genes. The discovery of oncogenes provided a simple genetic basis for the loss of control over the cell cycle that characterizes cancer. Even more important was the discovery of cellular counterparts of these oncogenes. These so-called proto-oncogenes are normal cellular genes that become oncogenic when mutated. As only one allele needs to be mutated, these genes act in a genetically dominant fashion and are gain-of-function mutations. Consider the example we have just seen of signaling by growth factors. Some proto-oncogenes encode receptors for growth factors, and others encode proteins involved in signal transduction that act after the receptors, such as the many protein kinase enzymes. If a receptor for a growth factor becomes mutated, that is permanently on, then cell division will occur in the absence of the growth factor. This is analogous to a light switch that is stuck on. The light will always be on. One simplistic way to think about this is an analogy to an automobile. Oncogenes function as the accelerator to push the cycle forward. Tumor suppression genes are also normal cellular genes that, when mutated, can lead to cancer. But in this case, both alleles must lose function to result in cancer. That is, these genes act in a recessive fashion. In an automobile analogy, this makes tumor suppressor genes the brakes. The first tumor suppression gene identified was retinoblastoma susceptibility gene, or RB, which predisposes individuals to a rare form of cancer that affects the retina of the eye. The role of the RB protein in the cell cycle is to integrate signals from growth factors. The RB protein is called a pocket protein because it has binding pockets for the proteins. Its role is therefore to bind important regulatory proteins to prevent them from stimulating the production of cell cycle releasing proteins such as cyclins or CDKs. The gene mutated most frequently in human tumors is the tumor suppressant gene TP53, which encodes for the p53 protein. This gene has been called the guardian of the genome because it is involved in the G1S checkpoint, where damaged DNA causes p53 to pause the cell cycle to give the cell time to repair the damage. If the damage is severe enough, p53 will cause the cell to initiate programmed cell death, known as apoptosis. When a mutation removes p53 function, the cell can proceed through the cell cycle with damaged DNA, leading to further mutations that lead to cancer. Due to its obvious importance, TP53 has been the subject of intensive research. This has revealed that p53 lies at the center of many intersecting signaling pathways and is important to integrate many aspects of physiology. The p53 protein responds to a variety of stressors, including DNA damage, and affects a variety of positive and negative regulators of transcription. The action of p53 can affect the cell cycle and apoptosis, but also inflammation and even metabolism. Thus, p53 has many roles, some of which lead to acting as a tumor suppressor. So here is figure 10.23, and it shows a number of key genes that have been found in the analysis of many different cancers. To end the chapter, whole genome sequencing. The classic model of cancer development is a series of mutations that move a cell in a linear progression toward malignant transformation. While this has not been invalidated, it has proved simplistic. Sequencing cells within tumors and comparing them with normal cells have allowed an unprecedented view of the genetic changes in cancer cells. Early results using traditional sequencing and mapping methods identified a large number of oncogenes and tumor-suppressing genes, leading to the idea of mountain genes, which are mutated in a significant number of tumors, and hill genes, which are altered less frequently. The application of next-generation sequencing technology to cancer genomes has allowed a much more detailed analysis of genetic changes in different cancers. New categories of genes have been identified: genes involved in DNA repair, mRNA splicing, and chromosome architecture. These genes probably act by increasing the number of mutations or changes in gene expression within a tumor. It is also now clear that different cancers show different spectra of mutated genes. This is important both in terms of diagnosis and in terms of more specific therapies for specific cancers. Significantly modified genes, SMGs, associated with specific tumor types are now being cataloged. These data will be combined with information on how an individual's patient's genotype affects both the growth of cancers and the drugs that might be effective for a specific tumor. The sequencing of individual cells in tumors has also revealed heterogeneity in tumor cells. This implies that a tumor is not just a clone of cells derived from a single transformed cell. Different cells can have different spectra of mutations, and there is variation in the amount and kind of chromosomal alterations in cells within a single tumor. This has even uncovered new mechanisms that damage genomes and chromosomes.